Hello everyone, my name is Tianyi, and today I'm going to give a brief introduction of our project, which is scDesign2, a high-fidelity ICR6 simulator that captures gene correlations. So first, let me talk about the motivation for this project. So in the field of single-cell RNA-seq, there are two main problems that need to be addressed. First is the experimental design problem. So how to choose among existing experimental protocols, like do we use a full-length protocol like SmartSeq2 or a tag-based protocol like 10x genomics? And secondly, given a protocol, how to determine the optimal experimental parameters. For example, we need to determine how many cells we need to sequence, the cell number, and also how deep we need to sequence, which is the total sequencing depth. And the second set of problem is regarding the computational method benchmarking. So as we know, in single-cell RNA-seq data analysis, usually there are many methods that have been proposed for the same type of analysis task. And how to choose among this available method is often a headache for the users. And our proposed solution is to use a realistic simulator to answer these questions. So the benefit of simulated data compared to real data is that, so first of all, they are easier and cheaper to generate than real data. And secondly, when simulating data, we usually have the ground truth. So there will be less uncertainty when evaluating the results. So regarding simulators, actually, a lot of them have already been proposed. So here we've listed some simulators, some existing simulators, and also our proposed scDesign2, and also some desirable properties of a good simulator. So first of all, we want it to be protocol adaptive, which means that it can simulate both full-length type of protocol and also tag-based protocols. And secondly, we wanted to preserve genes and then capture gene correlations. And then we also wanted to be able to simulate data with different cell number and sequencing depths, because this will help with the experimental design part. And then we want our simulation model to be easy to interpret. And then also the fitting of the model should be computationally and sample efficient. So as we can see that although many methods have been proposed, actually none of them can achieve all of the 6 desirable properties at the same time. But our proposed scDesign2, we believe that it can achieve all of the 6 properties. So here is a diagram of our proposed scDesign2 method. So our input is just a real data matrix, real count matrix with non-negative integer values. So for example, here is a gene by cell matrix, and we assume that the cells have been partitioned into cell types. And then for the model fitting part, we use the submatrix of each cell type to fit a joint distribution of gene expression for each cell type. And in the joint distribution, we not only learn the marginal distribution of gene expression, but also the correlation between the gene expression between different genes. And then in the data simulation step, we use our fitted joint distribution along with user-specified sequencing depths and number of cells to simulate synthetic data, which can be used for experimental design and computational methods benchmarking. So first, we can compare our method to existing simulators. So here we use a multi-cell type data set, and we compare its t-SNE visualization patterns. So as we can see that the pattern of data of scDesign2 is closest to the real data compared to the other simulators, and also our own simulator without modeling the correlations. And also if we mix the simulated data and the real data together and then do the t-SNE plot, we can see that scDesign2 achieves the best mixing compared to the other simulators. And then let me talk about the application of our simulator. So first of all, it can be used for other type of single-cell technologies other than single-cell RNA-seq. So here we have an example where we show the a t-SNE pattern of simulated data and real data for the merfish data set. So again, our scDesign2, the pattern of scDesign2 is closest to the real data compared to the other simulators, and also it has the best mixing. So finally, we can use our proposed simulator to help with the experimental design and combination method benchmarking. So in our manual script, we consider two tasks, which is clustering and rare cell type detection. So for clustering, we consider two experimental design scenarios. In each task, we consider two experimental design scenarios. So in the first scenario, we keep the number of cells sequenced as fixed, and then vary the total sequencing depth. In the second scenario, we vary the total. We keep the total sequencing depth as fixed, and then vary the cell number. So you can see that in the first scenario, as we increase the total sequencing depth, we have more information, and therefore the clustering accuracy will increase, as indicated by ARI and AMI. But for the second scenario, actually there is a balance point of optimal cell number, because here we're actually fixing the total sequencing depth, which is mimicking a fixed budget situation. So you can see that if we have too few cells, then there will be not enough cell to constitute each cell type. But if you have too many cells to sequence, then each cell will not have too many reads, and therefore we have a balance like this. And then so the method we evaluate are C-RAD and SE3, and you can imagine you can also evaluate more methods. So for real cell type detection, the analysis is similar. So the metric we look at is F1 score, precision recall, and so on for detecting a rare cell type. Okay. So here is a summary of our method. So scDesign2 is a transparent simulator that generates realistic single-cell gene expression count data with gene correlations captured. And we have an R package available at this link. And for future work, we want to extend the current model to accommodate continuous cell trajectories. Thank you for listening.